many members from the lab who have prepared really detailed explanations how to use Cas9 and other CRISPR systems for a variety of different research applications. There are many different ways to use Cas9. We can use it to target individual genes in the genome. So you can edit a gene, you can knock it out, put in a mutation replacement, or you can delete out a specific genomic segment. So those are the low throughput applications. That's what we'll hear first. We'll hear about the fundamental principles of how to design the system, how to use it for these individualized applications. After that, we'll have a second session to talk about how to use it in high throughput fashion. How do you use genome scale libraries to do loss of function or gain of function screens to be able to identify and discover a new gene function. And then finally, the field is still very young. It's only been, this tool has really only been used for a few years. The final part of the workshop will tell us about where the field is headed. What are the major challenges? How do we use research and development to be able to improve these technologies to make the application even more powerful? We'll have a number of folks from the lab who have been working on developing these technologies, and it is no one's better than them to be able to tell you how to actually use the tools. They are wearing, most of them are wearing green t-shirts with a Cas9 on the front. So feel free to find any of them during the breaks or even during the talks if you have a question that you really urgently want to have answered. So without further ado, the first speaker will be Nina Pazoka, who will tell us about the basics of Cas9. And then along with Lingyi and David Cox from the lab, they will tell you how to use it to generate cell lines, how to generate animal models for a variety of applications. So let's welcome Nina. Thank you.
So good morning, everybody. My name is Nina Pazoka, and I'm a graduate student in Feng Zhang's lab. Today, along with Lingyi Gao and David Cox, we'll be discussing CRISPR basics and using Cas9 for targeted DNA mutagenesis. So here's a brief overview of what we plan on talking about. We will cover an introduction to CRISPR-Cas9 systems and specifically go into Cas9, Cas9 nickase, as well as CPF1. We'll describe some online tools that we use that are really helpful for designing guides and using CRISPR. We'll also talk about how to process samples in the lab, how to read out these samples, and then if you're interested in doing homologous recombination, we'll touch on some of those topics too. So this is basic information and should be especially helpful if you're new to CRISPR or just getting started with it in your lab. I'd also like to point out that we will not be discussing certain things in this module. So we won't be discussing CRISPR-Cas9-based screening applications, RNA targeting tools, enhanced specificity tools, and AAV delivery in vivo. We will be covering these, but just in a later module, so stay tuned. So at this point, I'd like to turn it over to David Cox, who will introduce genome editing.
Hey, okay, so I'm David Cox. I'm also a graduate student in Feng's lab, and yeah, let's get into it. So if you guys are here, you're interested in genome editing, and as you know, genome editing is making targeted changes to the genome, usually inside of living cells or tissues. And the way that this has been mostly approached has been through manipulating endogenous double-strand break repair pathways. In mammalian cells, this really falls into two main categories that get manipulated during genome editing. In most cells, especially cells that are not actively undergoing mitosis, the way that double-strand breaks are repaired is through this process called non-homologous end joining, where two broken DNA ends are directly ligated back to each other. And if this process happens over and over again, eventually what will happen is you'll get a mistake that will happen in the form of a small insertion or deletion, commonly called indel, which often will result in a frame shift mutation and a premature stop codon, leading to complete knockout of that gene in a lot of cases. The other pathway that we think a lot about is called homology-directed repair. And in this case, if there's a piece of DNA that is homologous to the break site, endogenous machinery can use can interact with that homologous DNA and use that to synthesize DNA that gets incorporated into the original break site. So this is what we use when we want to insert, say, genes of therapeutic interest or GFP, and we'll talk about how you can specifically design that and manipulate that in a little while. So genome editing is based off of creating artificial double-strand breaks so that you can manipulate the genome at points of interest for yourself. And this has been done using programmable DNA nucleases. For a very long time, this was approached using technology called zinc fingers and talons, which are composed of a modular DNA binding domain where the protein kind of DNA interaction code was known, and you could string them together in such a way that they would recognize a unique sequence in the genome. And then this was attached to a nuclease domain. So the DNA binding domain and the nuclease domain were separate. In this case, the nuclease domain is FOC1, which has to dimerize in order to cleave DNA. And now what's become popular, and which is, of course, why you're here, is CRISPR, which is RNA-guided DNA cleavage, which is a little bit easier for most people to use. So when we talk about Cas9, it consists of two components. There's the protein, Cas9, and then there's the sgRNA, which stands for single guide RNA. And Cas9 has two domains that are DNA nuclease domains. One is called the HNH domain, and the other is called the RUFC domain. And each of these serves to cleave one strand of DNA at the target site, leading to a complete double-strand break. Then for the sgRNA, there are really two parts of it. There's the guide, which is the part that conveys target specificity, which is 20 base pairs long, and it begins at the 5' end of the sgRNA. And then there's a scaffold, and this scaffold is important because this is what's needed for Cas9 to really interact effectively with the guide. So when we actually get into how you use this to target a piece of DNA for cleavage, there's two things that are important. So I just mentioned the guide, right, which confers specificity, but there's this other thing called the PAM, which you've probably heard about before, which is also very important. And Cas9 uses protein DNA interactions to interact with the PAM, okay? And it must have this interaction in order to productively cut DNA. In the case of the very commonly used S-pyogenese Cas9, this PAM is called NGG. And the way that it works is you'll have a Watson-Crick base pairing between the guide sequence and one strand of the target sequence, okay? And then on the non-complementary strand, the strand that has the exact same sequence as the guide RNA, if the guide RNA was DNA, right 3' of that sequence, you have to have the motif NGG, where N is any nucleotide, and then you have the two Gs. Okay? And then once both of these interactions occur, most of the time what will happen is the HNH and the RUFC domain will come in and will cleave the DNA, creating a double-strand break as shown here. So something that was very confusing for me at first, and I think will be confusing for a lot of people, is if you have a target sequence that's on if you now flip it around and now you want to target something where the NGG is on the bottom strand, you have to change the orientation of the guide. So now the guide must be complementary to the top strand, and the NGG still has to be in the 3' orientation relative to the strand that has the same sequence as the sgRNA. And there are a lot of tools online to help you do this in case it's confusing to you. So there's also a nickase. And what a nickase is something that will just create a single strand DNA break. And this is achieved by mutating one of the domains in Cas9 that cleaves one of the strands. So it'll only cleave one strand at a time. And this is advantageous because single strand breaks usually will not result in non-homologous end joining. So most of the time, you can use this to very specifically achieve homology-directed repair. The downside is that this often occurs with a much lower efficiency, but yeah, that's something to consider. And then the other thing is, if you want to make this process very specific, you can use 2 nicases. So each nick on its own will not be converted into double-strand break, but if you have 2 nicks very close to each other, this will actually be converted into a double-strand break. And having the need to have 2 Cas9 sgRNA complexes successfully make a nick right next to each other really increases the specificity of the system, which is something that's would be of concern if you're making, say, a clonal cell line or an animal line. You want to make sure that you're just making a very targeted change. And so this can really increase the specificity of the system. S-pyogenase Cas9 that we've just been talking about is not the only tool that's out there. There are many different Cas9s, or Cas9 orthologs that exist, some of which have been characterized in great detail in mammalian cells and tissues. And so I just want to walk through really quickly what are the other technologies that are out there and how do they work. And so we were just talking about S-pyogenase Cas9. The PAM location is 3' of the target sequence, and the PAM is NGG. And this works incredibly reliably. If you pick a few guide RNAs, if you pick, say, 3 guide RNAs, most of the time you'll get one of those guys to work with pretty high efficiency. We also have S. aureus Cas9, which has the PAM in the same location, 3' of the target. Sorry, there's a mistake up here. The PAM is NNGRRT. And what's notable about S. aureus Cas9 is that it's smaller than S. pyogenase Cas9, so it can fit into AAV vectors. So if you have applications that require the use of AAV, this is a very good tool for you to use. And then we have CPF1, which was described after Cas9, and serves a very similar function to Cas9 in that it's a single protein that uses a single RNA in order to achieve target cleavage. But there's some things about it that are different and interesting. So instead of having the PAM on the 3' end of the target, the PAM's on the 5' end of the target. And instead of the PAM being 3' rich, the PAM is pyrimidine rich, in this case TTTV. And so this can be useful for targeting AT-rich regions of the genome, which is, of course, of interest to a lot of people. And the other thing to note is that the sgRNA is much shorter. So for certain applications, that can be a big advantage. And Lenny? Assay Cas9 is, I think, 1,000 amino acids. Is that right? Yeah. And then CPF1 is, I think, 1,200. No. Right. It's right around 1,200. And then S-pyogenase Cas9 is 1,300.
Thanks, David. My name is Lin Yi. I'm also a graduate student in the Zhan Lab. And there's many great resources on AdGene for CRISPR plasmids, anywhere from cutting, nicking, down to screening and visualizing. And there's a great compilation of many plasmids that have been deposited by many labs around the world that have been really useful to us and broadly useful. And I encourage you to visit this page if you haven't already to find the vector that might be the best for your experiment. So as David talked about, one of the most attractive parts about the CRISPR-Cas system for genome editing is that it's highly modular. Of the 3 main components of CRISPR-Cas9, namely the nuclease, the guide scaffold, and the variable region of the guide, only one of those components usually needs to be changed from experiment to experiment. And that's the short variable region of the guide that directs Cas9 to the right place in the genome. So that being said, even though it's the smallest component that we need to choose, it's also arguably the most important one. And in any gene in the genome, there might be dozens or hundreds of potential target sites with a targetable PAM, NGG. And how do we go about picking the best one? So I think one of the tools that's been really helpful is that there's been a lot of online tools that have been developed in the past 3 or 4 years based on experimental data about a large number of guides to help us pick the best guides. Here is a collection of them. And more recently, there's a review article that's out that has a really nice summary of all of them, including the functionality and the different pros and cons. But rather than going through all of them, I encourage you to you can check out this review article here and pick the tool that's the best. So as a quick preview, so an example of one of these tools in the previous slide is the MIT CRISPR design tool. This is something that was developed by our lab. So the way this works is that you can put in any gene or any target sequence that you're trying to target, and it will automatically generate all the different possible guides for Cas9. So here's the target, and the different guides are listed above. And for each guide, you can hover over it, and it will give you all the different potential off-target sequences that are very similar to the original guide in the genome, but have a number of mismatches, as well as how likely those off-targets are estimated to be significant. So for example, for this one, it's an off-target for this guide, 72 right here. With a score of 72. It gives you all the mismatches as well as the scores. So the ones with the highest scores are most likely to be cut. So using tools like these, we can pick a combination of the most efficient and also the most specific guides. However, our understanding of how guides work is not perfect yet. So a lot of these estimates are just estimates. So for example, I can't just take this off-target score 7.1 and said this will definitely be a more significant off-target than this third one, which only has a score of 1.1. The tool just isn't good enough to tell us that. It could be that the most significant off-target is actually down here, and right here, the first one is actually not cut at all. That sometimes happens. So it's important to actually experimentally validate these guides. So here's a timeline of, or an experimental timeline of how we would normally do this. So the first step is to use some of the computational tools that I discussed earlier to pick a collection of guides that we want to screen on or to test to pick the best one. Then we want to clone these guides into some kind of a vector, usually one that already has the guide RNA scaffold in Cas9. Deliver those into a cell line, and then either we can read out directly what the rate of editing is, or we can use individual cells in that cell line to create clonal expansions for downstream experiments. This usually takes, at least the first part, the first 3 steps, about a week, and then the clonal expansion takes a little bit longer for the cells to grow and to validate the cell line. So here are 2 of the ways that we like the best to test guides. 2 main ways. We can clone the variable region of the guide into a plasmid that already has Cas9, or we can synthesize the guide as a PCR product by putting a promoter in front of the guide. Usually the PCR product is about 300 to 400 base pairs. So the advantages of these methods are that usually by cloning it into the plasmid, you can get higher efficiency editing. And it's also a single construct you need to transfect. By doing it as a short PCR product, we transfect 2 different things at the same time. And this gives higher throughput if we're testing multiple guides, because it's easier to make this than to clone a bunch of guides into plasmid. I think for Cas9, the second way tends to be more expensive than the first one because you need to order a pretty long primer, as I'll discuss in a bit. And so I think for the second one, the main advantage is really just that it's easier and you can increase your throughput. If it's just testing a small number of guides, I would not use the second one. I would focus on the first one by cloning it into the plasmid directly. So here is an overview of the first method by cloning it into a vector with Cas9. So a lot of these vectors, including the one that our lab has put out, has, in place of the variable region of the guide, so there's a promoter, U6 promoter driving the guide and then the sgRNA scaffold. And in place of where the guide normally goes, we've instead inserted two consecutive type 2S restriction sites. And these are nice because they basically serve as a filler for what is actually supposed to be in there, but those are easily removed. And it's nice because the recognition site for these restriction enzymes is actually different from the cut site. So it'll recognize somewhere in the middle, and it'll cut out here. So all this stuff in the middle, it's only there as a boilerplate, and it gets totally removed once we clone the vector in. So if we digest this plasmid with, in this case, a BBS1, the stuff in the middle gets removed, and we're left with these sticky end overhangs on both sides. And now we can use this to paste in the guide sequence targeting the gene that we want right here. So really all we need to order is whatever insert actually gets pasted in here. So these typically are ordered as two different oligos or primers, as two different strands that are annealed together with the correct overhang on both sides that matches what the overhang should be in the actual plasmid. So how to design these oligos? I should point out that, so for example, if this is the genomic sequence with the NGG PAM here, we take everything up to but not including the PAM. So here is the target sequence, and we append the overhang that will allow this to be cloned in. Similarly, on the bottom strand, we append this overhang. Well, the CAAA on the other side, that's not the same as the PAM. So usually I will order about 24 to 25 base pair oligos, 2 of them for every guide that I want to clone, and anneal them together. So on our website, there are some protocols about how we go about cloning these guides. One of them is here. So we take the oligo, or the vector, and we digest it with BBS1. In the separate step, we anneal the 2 primers together to get the insert. And then we ligate the 2 pots together. This usually is very efficient cloning. When I do this, about 90% of the colonies have the correct insert. And there's usually several hundred colonies on one of the small ampicillin plates. So for improving cloning efficiency, some of the things to keep in mind are just to make sure the overhangs are correct and they have the correct length. Using a buffer with ATP so that the phosphorylation of the primers will go. And also maybe treating the vector digest with some alkaline phosphatase to remove phosphate groups to prevent the vector from possibly religating. So here's a variation of the same protocol that's a little bit less labor intensive, because if we use the observation that because these are type 2S restriction enzymes, the BBS1, once they get digested, the recognition site is actually no longer present in the vector. So once the ligation of the guide happens, that's sort of a terminal state of the reaction. Because of that, we can add the restriction enzyme and the ligase in the same pot without doing an explicit digestion. This is called the Golden Gate assembly. And we have a protocol for that also on the website. I think for me, the pros and cons of doing it via Golden Gate versus just via directly digesting and ligating are, this is a little bit less work, but it also takes a little longer to cycle the thermocycler for 15 cycles and to do everything. This may take about 3 to 4 hours to actually finish the transformation and plate the ampicillin plate. With directly digesting it, maybe it takes about an hour or so to finish the whole thing. So, So the second method is making the short PCR product and just transfecting the linear PCR product directly for the guide. For this one, we can design a custom primer that amplifies off the reverse strand of the U6 promoter and a standard U6 forward primer. And that will basically introduce, that basically concatenates the U6 promoter, excuse me, in front of the guide that we order as a primer. So how to design this? We concatenate in the 5' to 3' order the U6 priming site, the guide, and the sgRNA scaffold, and reverse complement the entire thing. And this usually results in something that's relatively long, so you need to order it as a special ultramer in order to get the right size. And the amplicon, after amplifying it, would be about 350 base pairs. I usually try to run it out in the gel to check that I have the right thing. Ordering it as ultramer actually is kind of expensive. So one alternative way to do this is to order the entire thing as a G-block. That's a little bit gratuitous, but I think the cost actually is about $89 each. So if it's really a throughput thing, that might help as well. For transfections, here's a typical transfection day. So the day before, I'll plate the cells, about 120,000 cells in the 24-well, each well in the 24-well dish, and try to have it about 70% confluent when I transfect it. I found that one of the most important factors influencing transfection efficiency is how confluent the cells are. So if they're like 95 or 100% confluent, then the rate of editing drops significantly. So about 70% is the sweet spot. On the day of transfection, they deliver either 500 nanograms of the PX330, the Cas9 plasmid with the guide cloned in, or if I'm doing it separately, if it's a PCR amplicon and a Cas9 on a different plasmid, then 400 plus 100. We've done time course experiments to see how fast the editing happens. And usually after about, at least in hex cells, after about 2.5 days, the editing reaches saturation. And I can harvest or clonal expand the cells anytime after that. Are there any questions so far? OK, great.
All right. So Lin Yi just gave us a good description of how you use CRISPR in the lab. And so once you transfect your cells or you have CRISPR in your guide in the cells, you want to see did it work and how well did it work. So there's two different ways that you can read out and to see how well your cleavage worked. And the first one that we use is called the surveyor assay. And the good thing about surveyor is that it can be done in any lab with basic molecular biology technology. And the surveyor assay, overall the way that it works is that it takes a pool of PCR amplified genomic DNA, it anneals it back together and rehybridizes it, and this particular surveyor nuclease enzyme recognizes these sites that are annealed that are mismatched and cuts them. And so let me go into a little bit more detail. And so after transfection, you isolate your genomic DNA and you design primers spanning the target sites. We recommend between 500 and 800 base pairs. That's just based on what we've done in the lab. That works the best. And then you PCR amplify this genomic DNA. And then it's important that you clean these up, and you can use a QIAGEN kit, for example. And then this is the key part of the assay. You melt and re-anneal slowly in a salt-containing buffer. And so what happens is, let's say you have 3 amplicons, and when you rehybridize them, it's possible that some of these indels can rehybridize with a wild-type version. And so this indel is mismatched with this wild-type version. And again, that surveyor nuclease is able to recognize those and cleaves those fragments. And then after this is cut, you run it out in a gel and you quantify it. And so here is what surveyor looks like once you run it out on a gel. And so this band up here is your uncut fragment. And if you look here, so that's the uncut, the long version. And then you will produce two smaller fragments, fragment A and fragment B. And you'll notice that if you add these two up, they equal the size of the PCR product. And then to quantify, to see how well your Cas9 worked and how efficient your how efficient the indel formation was, you can use this formula here and gel quantify these bands. And so when using Surveyor, the most important thing is that you have a very clean PCR amplicon. And so to troubleshoot Surveyor, the main thing that we recommend is that you do basic genomic PCR troubleshooting. And so the first thing is if you have multiple bands or you don't have a PCR product, we recommend that you try varying the TM. It's also very important that you use, for example, magnesium chloride, because this is a genomic DNA reaction, and if you're working with a large genome, then this will help make a clean band. The reason why having a clean band is so important is that if you had multiple bands here, and especially if they were similar sizes as these fragments, it might make it more difficult to quantify how well your indel formation occurred. Other things you can do for optimizing your PCR is try different polymerases, try different primers, different primer TMs, and then if all else fails, just redesign your primers. Another important thing when working with Surveyor is that, again, your PCR band needs to be clean, and that if you have smeary lanes in your gel, this can also make it more difficult to quantify. And so if you're seeing this, possible reasons are that you have residual ethanol from the PCR cleanup. You could have nuclease contamination. And then another key thing here is the Surveyor nuclease is pretty active, and the protocol recommends that you only digest for an hour. And so if you leave this over time, if you're out for a while, if you over incubate, this can lead to smeary results that that aren't good, and it'll be difficult to quantify. Another method is just using NGS and next-gen sequencing. And this is especially nice because it's very high throughput, and you can test multiple different samples at the same time. And it's pretty easy, too, because all you need to do is just, you'll do 2 rounds of PCR. The first round just amplifying your genomic region, and then the second round where you just add barcodes. And then you can pool those all together, run them on an NGS, and then during the analysis, you can see the indels, and you can actually see if there were insertions, deletions, and everything in between. And so just a brief overview of the 2 main methods that we use to read out indel formation. So the first is Surveyor. The great thing about Surveyor is it's super quick. You can get results in the same day that you harvest genomic DNA. But it is really best, it's much more low throughput, so we recommend even 15 samples or less. You don't get any sequence information, you just know if there was an indel or not. But you don't need access to any next-gen sequencing machines. Whereas with NGS, it takes a little bit longer because you need to prep the library, run it, and then analyze it. But you can do as many samples as you can. You can load tons of samples on here, so that can be really nice. Also, you can actually get sequence information, seeing what's being inserted and what's deleted. But again, you need access to a machine. So any questions on this readout before I move on to HDR? Great. Okay, so one big application for using CRISPR is to do homologous recombination or introduce specific mutations, or, for example, GFP fusions or knock-ins. And CRISPR-Cas9, as we've described, is really good at cutting DNA at a very specific site. And by cutting DNA, that's a great way to initiate homologous recombination. But as David was saying, the main way that many cells repair broken DNA is through non-homologous end joining. HR is another form. And so HDR is not nearly as efficient as producing an indel. And there are a few tricks that you can use to make this more efficient, but overall it just takes a little more time and more screening. And HDR efficiency, as you may know, it really varies by the cell type. And so it might, for example, mouse ES cells, it works quite nice, whereas other cells, it's very difficult, if not impossible, if they're not replicating. And so when doing HDR, again, you use CRISPR to introduce the double-strand break, but you also provide this template. And there's two different ways that you can provide a template. So first, you can use a plasmid-based system, and then you later linearize the plasmid. And this is good if you want to make a long modification, for example, GFP knock-in or something like that. Another thing you can do if you need just a very short introduction, or if you're doing like a SNP change, for example, is you can use like an ultimer. And that's nice because you can order these from IDT. It's really easy. And in general, it's higher efficiency, but again, this will really vary based on your cell type and your target. When designing this template, you want to have the change that you're going to make or the addition that you're going to make, but you also need to have homology arms. And these homology arms, depending on where you look, will cite different lengths. And so if you don't use any nuclease, previous papers have suggested really long arms, for example, 1.7 kb. If you are using CRISPR, something that our lab has recommended, or I've generally heard, is a general recommendation of 1 kb on each side. But again, you can try different sizes. If you're doing a single-stranded oligonucleotide, you can have as small as 30 to 60, as seen with Cas9 and C. elegans, 80 base pairs. And so these can be relatively short, but they also do work. If you're making a plasmid, there are a couple different ways you can do this. First, you can just buy a G block, but this can be a little expensive depending on the size of the construct that you're making. You can also get cDNA and then modify your plasmid. Or lastly, you can just collect genomic DNA, PCR it up, and then introduce it into a plasmid. And again, when you're working with the plasmid, it's important that you linearize the plasmid when you're using it with HR. Okay, so again, when you're doing HR, you use CRISPR to make the cut, and then you have your repair template. And as we've discussed previously, you can, CRISPR or Cas9 is great, especially as pyogenes, because it can target, there's so many different sites that it can target within the genome. However, when selecting your target that you're going to cut when you're trying to do HR, you need to take a few things into consideration. So first, it needs to have a PAM site. Second, you ideally want it to have low or no off-target cutting. And then lastly, especially for HR, it's recommended that you do about 30 base pair away from the side of the double-strand break. So it needs to be in pretty close proximity to where you want to modify. Just some practical tips, if this is new to you, something that we've found very useful in the lab. We found that it's really nice because HR is a bit lower efficiency to have a nice way to assay this and to see if HR has occurred. And so one thing we recommend is that you can use, for example, synonymous codon mutations to introduce a specific restriction enzyme site. And that can be specific to your knock-in. And then you can attempt to do HR and then get a pool genomic DNA, PCR it up, and then test to see if you get a cut site. And so that's one way you can, is a pretty easy assay to see if HR worked at all. And so here's an example of kind of the things I've just talked about to put it all into context with an experiment. And so let's say you want to knock in this piece of DNA here. And so if you're using the plasmid repair template, again, we recommend about 1 kb arms. And we've introduced a restriction enzyme site in the center. Or you can also do a single-stranded oligonucleotide. either the sense or the antisense. That will both work. And so you can attempt HDR, and then PCR your genomic DNA, and test to see if you get any cleavage. And this is an example of how you can design your target for cutting with Cas9, and then where the HR is occurring. And so you can see that, again, they occur in close proximity. And here's a readout. So for example, in hex cells, we were able to get pretty good HR efficiency, up to 27%. We think it's pretty good. Whereas in Hughes 9, which is a human ES line, much lower efficiency, the highest was at 6%. So it is possible. Again, it'll really depend on your cell line. And this is a good way to screen if HR is occurring at all. And so since HR or HDR is a pretty low efficiency process, you can try to enhance it. And papers have shown that if you suppress the NHEJ protein, so the other pathway to repair a double-stranded break, it can push the repair mechanism towards an HDR method. And to do this, you can do shRNA gene silencing of these NHEJ proteins, or there's also a known ligase 4 inhibitor, SCR7, that you can use, and people have found that's been helpful. So are there any questions on this that I just talked about, or anything related to this presentation? And if you want to talk, you have really particular questions about a certain experiment, we invite you to find any of us in the green t-shirts, and we can help answer your questions. And so at this point, I think we'll have I'll turn it over to Rhiannon.
Okay, thank you, Nina, and thank you also to David and Linyi for going over the basics. So what we wanted to do next was basically just have an open half an hour where you guys can get one-on-one help. I assume that most of you guys came here because you have experiments in mind that you want to start using CRISPR-Cas with. So there are a number of us up here in the front row. Most of us have these green t-shirts on, but there's a few other people. So we can just kind of walk around, and feel free to come up to us. But this is, I mean, if anyone has any questions they just want to ask openly, that's fine. But if you want kind of like tailored one-on-one, we are also available for that. Okay? So ask us whatever you want.